This video is on Plant Derived COVID-19 vaccine by Dr. Joseph Ordog, MD. It was not funded by any outside source. There's no external conflicts. Self-funded by myself. This was presented at the American Federation of Medical Research in Washington, D.C., April 2nd, 2021, and was published in the Journal of Investigative Medicine, April 2021. It was preprinted in ResearchGate online in 2020 and presented at the American Federation for Medical Research following this in 2021 on April 2nd. No conflict of interest noted. This was self-funded. Here we see initiation of the research. A coronavirus is identified by electron microscopy and serological tests as SARS-CoV-2 on the left-hand photograph. The right-hand photograph was a plant-derived virus-like particle that is possible investigation for a vaccine. Here's a photograph of the cell lysis experiment caused by the virus. The section A is the cell pasture, that is a thick coating of cells that can become infected by the virus. B is the lytic lesion of each viral particle as it forms a colony. As seen similarly with this rhododendron leaf under 1,000 power light microscopy, the similar lesions as the viral cell culture media would show, and this was used as a plant live culture media. This is the aspen leaf with a similar viral infection, and each of the yellow circles are actually viral colonies, and they've been tested out to show the SARS-CoV-2 virus in each case. Here's a similar situation with the salmonberries infected with SARS-CoV-2 virus. With self-attenuation, there's a symbiosis through the plant's immune system that keeps the plant alive and also keeps the viral colonies alive. A similar situation with the blackberry shows that the leaves are infected with SARS-CoV-2. Each little round colony is present on the leaf and identified, and with mRNA serology can prove that this is SARS-CoV-2. Here's their best subject, was the vine maple leaf infected with the SARS-CoV-2. The best vaccine media of all plants tested, the safest we felt, and it was a symbiotic relationship. The plant stayed alive, the tree did not die, and this seemed to be the least toxic of the materials. Here's the research presentation in Washington, D.C., April 2nd, 2021, showing the start of an abstract on this research article. Presented by myself, Dr. Gary Ordog, again on April 2nd, 2021. Here's the complete abstract, published in the Journal of Investigative Medicine, April 2021 again, with several of these same photographs from the research used in the article. This was peer-reviewed, accepted for publication, and did appear in the journal April 2021. Again, here's the cell lysis experiment, a little more detail on that. Each number B, the darker lesions are a actual viral particle that has taken over the cells on the cell pasture and increased in size, multiplying to form the cell colony. So each one is an original one virus cell colony. Here again is the EM, electron microscopy, in this case transmission, EM of the coronavirus SARS-CoV-2. On the left, the synthetic plant-like viral particle that is created in the lab. Here's a documentation of the plant's immune system, and it shows that it is quite extensive. Most physicians, such as myself, before starting this, did not realize that plants had a sophisticated immune system that they call the innate immune system, and it can also respond here to viruses, bacteria, even insects can be controlled by the immune system of the plant. And this is one diagram showing how that works. Another diagram showing the mRNA being taken in by the cell and how it is processed by the immune system of the plant to destroy or immobilize or attenuate the viral mRNA. Here's a scanning electron micrograph of SARS-CoV-2 that causes COVID-19, and it's been colorized. The viruses are colorized, the surrounding cells are not, or actually they're blue. Okay, so the first hypothesis that was entertained was that the virus is attenuated by the plant using its innate chemical immune system. Similarly, Pasteur used chemicals such as phenol to attenuate viruses and produce vaccines by that matter. The second hypothesis that the plant's immune system produced antiviral agents including lectins, flavonoids, and others, which limited the control of the colonies and prevented death of the leaf and the whole plant. And this was proven in our experimentation. The next hypothesis was the plant on a popular hiking trail by observation was exposed to human hikers, one of which expelled the virus onto the plant and infected it. This was observed, measured, and tested by serology. Another hypothesis, the plant chemical immune system, including antivirals, could attenuate the virus to allow both the plant and the virus to survive and grow. The virus, though, was attenuated and could be used for a vaccination. Medico was already growing the virus on tobacco, the production of an experimental vaccine, and this was one piece of evidence that the virus can grow on plants. We've already known that many viruses grow on plants. The first ever virus was the tobacco mosaic virus. There must be a cross-kingdom transmission of these viruses for this to occur. This was a hypothesis and completed with observations as noted in this experiment. Another hypothesis that both plants and animals can contract the infectious virus, and this also was proven both in this and other previous experimentations. The next hypothesis that the plant's chemical immune system prevents the virus from killing the plant because it's not completely efficacious. It does allow for a low-grade viral infection for the virus to multiply slowly, and this also was observed. It's basically a symbiotic relationship. Next hypothesis, that the self-attenuation of the virus occurred by the natural culturing of plant leaves that produce antiviral agents as part of their, quote, immune system, unquote. And this also was observed. And as previously documented on previous published articles. The next hypothesis was a symbiotic relationship developed between the plant using its chemical immune system and the virus, which allowed it to replicate in an attenuated fashion to allow both the virus and the host to survive and grow. The next hypothesis is that the non-toxic plants such as the vine maple, Acer cincinnatum, could be used to produce an oral plant attenuated vaccine. And this was our main hypothesis resulting from this experimentation. As the vine maple species, Acer cincinnatum, is non-toxic and safe, could be used to create an oral vaccine. Please press subscribe if you would like to continue on with further in this series. The next hypothesis involved the maceration of the infected leaves from the maple leaf, mixing with a non-toxic adjuvant and flavoring to promote assimilation and palatability, with the proposed route of entry being orally, as the SARS-CoV-2 appears to be passing through other animals. This plant source vaccine could be mass-produced to be introduced into the feed of the animals susceptible to the virus, including, but not limited to, non-human primates, minks, cats, etc. Further hypothetical studies may involve the production and controlled growth facilities, RNA sequencing, animal testing, and finally clinical tests, followed by phase testings in humans. Disclaimer: the information should not be construed as to currently engage in these methods as proven medical therapy. Further research is needed before recommending it. This, again, another statement from the lawyers, and a nice model of the SARS-CoV-2 virus behind that. This actual electron microscope views scanning EM of the SARS-CoV-2 virus. And further credits, Gary Joseph Ordog, MD, the author of this study and publication and video. To see more videos like this, please press my subscribe button, or thumbs up as well is nice. Recommended to others is nice, and comments of a positive nature would also be much appreciated. Thank you very much. This is Dr. Gary Joseph Ordog, MD, creator of this vaccine, signing out. Thank you.